Hello everyone. My name is Keith Parent. I am the CEO of Court Square Group, and this is the Driving the Business of Science podcast. We started this podcast just because I know that I work with a lot of different people over the course of my job, and they do a lot of interesting things that aren't necessarily developing drugs all the time. And I thought it would be good to have a podcast that just talked with those people about what they do, how they're influenced by this industry, and what they do to influence this industry. Court Square Group is a managed service firm. We manage IT infrastructure for life science companies. We manage infrastructure all the way from preclinical through manufacturing, focusing heavily in the clinical and regulatory space. And we've also got a product called RegDocs 365, which is a content repository that hosts clinical and regulatory content. Started out with the TMF and EDM reference model, and we've since created what we call Life Science Launchpad, which is a starting point for a lot of life science startup companies to be able to have good governance right from the very get-go. It's a pre-configured departmental folder structure with a governance plan. Today, we have a special guest with us, Dr. Joe Jerry. I've known Joe for a very long time as the executive director of HistoSpring, PVLSI, all sorts of other names of things that he belongs, and he's a researcher at UMass Amherst. So he's going to talk about a lot of these different things, and I'm very excited to be able to talk to Joe about what he's doing. So I'd like to introduce Joe Jerry. Joe, could you just give a little background on yourselves and tell us what your official title is at all those different places?
The official titles might take a while, but other
Now,
than
don't worry about it then.
wearing too many hats. But first, I just want to thank you, Keith, because this is an opportunity for me, because you really provide that third leg of the trifecta that's important. There's clinicians and there's scientists, but business, if we never bring it to a marketable standpoint, we're never going to be successful in bringing drugs and new therapies, new tests to the patients. So I really appreciate your efforts to educate, reach out, and bring people together. So I guess I'll start chronologically. I'm a faculty member at University of Massachusetts Amherst, a professor there, and I am in the Department of Veterinary and Animal Sciences, but all of our research has always been about breast cancer and in the biomedical space. And so in 2010, I became the science director at the Pioneer Valley Life Sciences Institute in Springfield. That's co-owned between UMass and Bay State, but it's a 501(c)(3), which allows some unique opportunities and flexibilities for us to interact with other organizations. And we've also, within that structure of the Pioneer Valley Life Sciences Institute, we've created other structures, the first being really taking advantage of the access to human tissues, and it created HistoSpring, which is a histological services core serving outside interests as well as Bay State at Medical Center and also UMass Amherst faculty, but really some of the colleges in the regions. And the other is that we created a patient registry, breast research registry, which combines a variety of resources, and maybe we'll get to that later.
That
But
would be great. Yeah, I definitely will want to cover that as a topic because I think that is a unique thing that PVLSI has to really be able to talk to people about that. So let's talk a little bit about PVLSI so people understand what that's even all about. I'm actually on the board of PVLSI. I'm one of the few people that is neither employed by Bay State or by UMass. So I can say whatever I want when I go to those board meetings, and it's great.
and people listen too.
I don't have to worry about my employer, which is a nice thing. And it gives me an outside look into what goes on across different institutions like that. And I've learned a lot about just clinical research and the different research these guys get, the NIH funded scientists and how they work. And it's incredible when you start to see just the amount of effort you have to go through to remain a scientist. And I don't envy you when you're doing that type of work, Joe, but I will say that when you do get the grants, it's a great thing for you guys to keep going and be able to give you that leeway to do what you need to do to kind of put things together. Tell us a little bit about that whole process and about PVLSI and what made you think about PVLSI that back in 2010 when you started.
So I've had the good fortune to have good clinical collaborators. So I've always felt, whether as a scientist or as a grant reviewer, you have to understand the disease you're working on intimately. And the pathologists, the oncologists, the surgeons have been there to really educate us and made it much clearer some of the things that we really need to get the goals that we have to have for ourselves and for our research. And so part of that was a natural extension was this Institute for the Pioneer Valley Life Sciences Institute, which was founded in 2002. And then I joined as the science director in 2010. And it was an opportunity to really bring those together and get some of the interactions with clinicians that I had the good fortune of having, make that more available to scientists at UMass. I can speak to our worldview as a scientist, that of a clinician, and that of the business world. These are overlapping Venn diagrams, but oftentimes it's a very small overlap, which we really need to fortify. And so when Keith speaks up at these Pioneer Valley Life Sciences board meetings, he brings a very different perspective, and everybody's suddenly, oh, I haven't seen it from that perspective before. So it's an important community to have. So it was really that opportunity to really bring together the research community as well as the clinicians to have more conversations, as well as bringing in that would like to really be the platform for launching new technologies, new opportunities, new scientific grants and investigations.
Well, when you mentioned that it was started up in 2002, I don't think that people really realize that Senator Ted Kennedy, was instrumental in helping get the funding to get PVLSI going. And over the years, the Mass Life Science Center has done a great job at helping to fund some of the opportunities that happen out of PVLSI. One of the things that I think that people don't realize is it deals a lot with, or it was meant to deal a lot with, translational research. And when we talk about basic research, we talk about, the large scale universities, and UMass Amherst is one of those. And the fact that Amherst itself is about 20 miles or so away, up in Amherst, away from Springfield. PVLSI is in Springfield. PVLSI is right on the main campus of where Bay State Medical is. Not necessarily the main campus, but it's close enough where they can bus up and do all that kind of stuff. Talk a little bit about what translational research is versus basic research, Joe, so people understand that and understand why we need translational research and why that's so important.
Uh, Personally, I'd define translational research very broadly. It's trying to build the information that you need to activate a new treatment, a new strategy. And that often means that you're working in model systems that are maybe far, far away. I spent a lot of my career working in mouse models of a syndrome called Li-Fraumeni syndrome, that you inherited susceptibility to a lot of different cancers. But it turns out that breast cancer is one of the main uh, ones that is, if you carry this inherited mutation, then breast cancer is your most likely tumor type. Fortunately, that's not a very common uh, genetic variant, but it is important to learn. And along that way, we learned that, surprise, surprise, mice are and are not great models for people. There are some wonderful things that we can learn from them, but we tend to work in single strains of mice that are very similar. And we know that a population of people is very different. And so when we looked at the effect of this mutation in one strain of mice, it had a very different response than in a different strain of mice. Literally, one strain of mice had mammary tumors, breast cancer was the most common phenotype, like humans, but the other strain had none. It tells us about the importance of that genetic differences among individuals that can render you refractory to even this really highly penetrant mutation. And so it gives you an opportunity that there must be some compensatory pathway. Then how do we study that in people? Well, we need to have access to clinical specimens and people. So it's a natural progression, I think, that we tend to go through. And so, this translational sciences is trying to just take information, observations, and really know what you want to get out of it to help patients. So it really has to start with that understanding of the disease, I think.
Well, it's interesting when you talk about that, Joe, because when you think about different mouse models and the fact that some mice show things and others don't, when you think about people and the political climate the way it is today, and we talk about DEI initiatives and things like that, the fact of the matter is in clinical research today, a majority of those people in clinical trials are white males. And the issue we have is that there's a lot of diseases that affect different ethnic groups and different races. And if we don't start to study those more, have those people involved in clinical trials, it's a whole different ball of wax when you're trying to deal with the drug's effectiveness and how things work. Luckily, there's a lot of people out there trying to work right now with looking at data over different patient populations, trying to find different capabilities across that. Let's talk a little bit more about that breast registry you mentioned a little bit earlier in the biorepository. I know that over the years, some of the different things that we spoke about with the breast registry and the biorepository, there's a huge opportunity there for looking at data a different way. Talk a little bit about that and how that works.
Well, so the opportunity is really what you were just talking about, in that if you're just looking in men, do you know what the reality is in people? Uh, and so if we often think about the equity and diversity as a disadvantaged population, but within this diversity that is the human race, there are some people that are going to be highly susceptible to either a environmental factor that can cause a disease or very resistant. Well, oftentimes it's that small population that is really resistant that gives us the idea of how we can take those that are susceptible and make them resistant, just like that subpopulation. That may be in a very a population that's ethnically quite distant from the Caucasian population. And so it's actually to everybody's advantage to widely sift across that pool of the humanity that we are, so we can understand those gifts and strengths that each population offers to us. So in the breast registry, we try to capture a reasonable diversity of individuals, and Springfield's not a bad place to do that, surprisingly. There's a cultural diversity in the patient population. Among the other things that's really fascinating is that, unlike some of the major medical centers in Boston where people come in for their treatment and leave, the typical patient stays in the Springfield area and at Bay State Medical Center for upwards to 25 years or more. So that means you have a clinical history on these people that is incredibly valuable. And so, as part of that breast registry, we do take advantage of that. We ask them a questionnaire that's modeled after the Nurses' Health Study, and enough information so that we capture what their background is, either educational, work, exposures, where they've lived, and what's their family like. So we have that questionnaire information, as well as one of the requirements is that we must have their biospecimens that are that come from either their clinical diagnostics and or surgeries, if they've had surgery there. So we have everything about this population of people. And I think the fact that they are in one place for extended periods of time, they're not moving around quite so much.
Well, and there's a couple of things that are advantages to that, right, Joe? And the fact that, I've known over my time with PVLSI, one of the things that we've touted is the fact that you may have multi-generational capabilities, looking at mothers and daughters and grandmothers and seeing how that affects that, and then how important that data is to looking at what happens, particularly in a family setting.
Absolutely. That if you're talking about any heritable disease, of which most of the diseases we're talking about, 30% of risk is inherited. We don't know the names of the genes yet, but your family makes a difference. And you can learn something from that. And the fact that we do have the possibility for multi-generational interactions with the patients makes it especially valuable. I should also mention that most of the patients, they can opt in or out, but more than 90% that are part of the breast research registry have allowed us to recontact and update their clinical histories as over time. So it is really a deep pool of information because they are in one place for decades.
Now, isn't there a possibility? Don't you guys have um um biorepository specimens that are of healthy cells as well as the ones that have disease? Tell me a little bit about that.
So as we went in, there are a lot of breast cancer resources through NCI support, National Cancer Institute supported uh, sites, but they've not focused much on normal breast tissue. And so part of our work with mice made us realize normal is something that is it has pretty broad range of what normal is. And so when you get back a blood test and you find out that you're out of range of normal, but doesn't mean you're abnormal. It just means you're a little different. And so that we try to make sure that we have a population of people that is representative of that diversity of, of susceptibilities and resistance.
Now, are you finding there's other people that are very interested in that? Do you guys use that data? There things that you do just for your research, or is it opened up to UMass researchers? How does that work?
So we've actually collaborated. It's open to everybody. It's not just us. And we've collaborated with people at UNC, University in North Carolina, as well as elsewhere, the NCI as well, to look at normal breast tissues that we have from these women, understanding there are certain important exposures. One is how many pregnancies have you had? At what age? When you started your first exposure to ovarian hormones and so really puberty to when you stop at menarche, stop cycling, those are really important. We captured that information and we've done looked at transcriptional profiling of how these tissues look differently at a molecular level. And it's been enormously helpful to understand as we parse out those women that they're from their normal breast tissue, how those life exposures affects that tissue to render it more susceptible or more resistant to breast cancer.
So I think, in the spirit of this podcast, when we talk about the business of science, you start to think about the fact that not only is it data you're collecting from patients, but it's also specimens that you're going to keep for a certain period of time that you can then use for further research and do more things with that. And I think it's important that people realize that there's many ways that they can help science and help to drive that. So having people that are open to doing that and being able to do it, particularly the multi-generational research and things like that that you're already talking about, I think that's hugely important. Talk to me a little about the branding effort around HistoSpring and how that works. What is HistoSpring? How is that involved with PVLSI? How does that all work?
So we've, I guess there are two questions in there that I want to address. And one is that about how the breast registry is under, if you look on our website, we've marketed this under HistoSpring. And so the access to the human tissues are within the HistoSpring. Now, trying to uh, provide access to these tissues there's only so much of it, oftentimes not very much. And so we have to think of ways to use those to the best extent. The normal breast tissue, we tend to get a larger amount of that because the tumors hopefully are quite small compared to the normal tissue within which they lie, or there's a contralateral mastectomy, removing the other breast. And so that gives us a lot of material that we can save and therefore can provide to different organizations for studies. And you mentioned about how what it takes to build that registry. One is quite, and not only what it takes to build it, but having people interested in donating to it. You can imagine, especially this day and age, donating your tissues and personal information, like a bit about your history, can be a bit forbidding. And I will say, my hat's off to the Rays of Hope and the walk that we have every fall, that there are close to 20,000 people involved in the Rays of Hope foundation that's partly through Bay State Medical Center. And these people are leading the way, not science. They are leading the way saying, fix this. What caused breast cancer? Why me? And they are offering their tissues and their information. And this requires an incredible amount of trust. And that reflects upon our success, really reflects upon the whole clinical team at Bay State Medical Center, the oncologists, the pathologists, the surgeons that have to participate in this as well. And trust me, there's no remuneration to the patient really, to any of the clinicians. And I'm just thankful to all of them working together because they realize this mountain we're climbing, we really need to do it together to really succeed. I should say too, we're talking about personal information. There are a lot of ways that we protect the patient's identity in this. So there may be patient specimens in a research study that's published, but it's always a little bit small amount of information that doesn't not enough to trace back to the individual. And it's also protected in a secure system called REDCap. REDCap was founded in 2004 uh, down at Hopkins, I believe. And we established the registry using this in 2010. So at that point, everything lives in a secure environment in the cloud. It's not on somebody's laptop. Even when I'm searching the registry, I don't know the names of anybody. And it's the data manager that really does all of that. And so we build these barriers to protect the patients, but it really, you can tell somebody that if you don't trust them that you're going to protect me, then you really don't have a registry. And so even the minority populations have been really generous and entrusting their specimens to us and
So
hope.
let's talk about HistoSpring a little bit more about the concept of when we put HistoSpring together. One of the big things was it's already being used for research, and Amherst, UMass Amherst can use it to do some histological services and things like that. Bay State can use some of that. Where else? I mean, you've opened this up for a much wider audience. Tell me a little bit about that and what your hope is with there.
Well, HistoSpring, our actually marketing is a relatively new strategy that it was pretty much an in-house project for Bay State, UMass, and some people collaborating with us that came to us. But by really building an online presence of HistoSpring that allowed us, even though it's still within the Pioneer Valley Life Sciences Institute, branding wise, HistoSpring's a lot easier to say. So we have brought everything under that umbrella for our web presence. And that includes the access to tissues, the histological services, the registry and background on the patients within that. And so hopefully we can start making this much more widely available to industrial partners as well.
Well, I think part of that is that, people out in the Boston area don't really know that much about what goes on in Western Mass. And, as far as they're concerned, Western Mass starts in Framingham. So it's one of those things. It's kind of a pretty closely held secret that we're out here with all of the work that you guys do and are already providing for people. So, we can open that up to a much wider base if they just know about it. So I think it's great that you're out here talking about that and putting things out there. And you're also tied in with IELTS over at UMass Amherst. Can you talk about that tie-in as well? Because that's the Advanced Life Science Institute over at UMass.
Absolutely. So reaching out to Eastern Mass, the Mass Life Sciences Center has been really good about providing some resources as well as building connections. And as I spoke about earlier, is trying to build that translational pipeline. The Pioneer Valley Life Sciences Institute was founded in 2002, and we really transitioned in 2010 to a different format where we were providing resources and building this registry. Along the way, the Institute for Applied Life Sciences at UMass has been modeled in many ways upon that experiment that was the Pioneer Valley Life Sciences Institute with a much larger um, support and funding from the state to build a large building that has a lot of research cores. So we have a lot more facilities. And so that brings opportunities where people want to get access to our tissue specimens, and then they need to do mass spectrometry. Well, we can do the microdissection of just small pieces of human tissues from formalin fixed materials oftentimes, and then provide them to the cores at UMass to do proteomics on a large scale, and a lot of other cores that are available through UMass. So it just adds to that continuum of the translational biosciences.
That's great. Can you talk just, we're going to wrap up in a few seconds here, but I want to just talk about what's the future? What are you looking for? What are you looking to do to grow PVLSI and HistoSpring and everything else that you're working on?
Well, make people more aware of the resources that are there. And so it's slowly but surely that's been happening. And really, we've only been trying to market seriously in the past 3 years. And so we're still developing that. I'm learning a lot from the help we've gotten from marketing.
That's
And
fantastic.
and so what we really look forward in the future, in 2022, the Biden administration had really put together this effort to look more at human specimens. Mice and model systems are great, but we really have to understand humans. Our genomes are enough different from those of other species that we can learn a lot of how to do it, but we don't know the exact targets. So we really need to work with those human specimens. And it's the so-called new approaches and models for using human specimens in research is a great effort that's being continued now. And so really starting with those human tissues where we have been able to collect a lot of normal tissues and cells from both diseased and normal breast tissue, turning them into models that we can grow in culture. And so really understanding how that individual responds to an environmental contaminant. We hear a lot about the environment, which is a huge part of the risk for any disease. And yet most people exposed to a compound are unaffected, but a subset really are sensitive to it. How can we identify those? So using those models in cell culture models, three-dimensional organoids, and also then adding back, you're dealing with just a ball of cells often in the organoids. Can we add back the immune system to those? And so building those in vitro models from diverse patients and people is really the goal for the next century, the rest of this century, I think.
That's awesome. I want to thank you, Joe. We're coming up on time here. It's been a fascinating conversation. I think it's great to hear how, a healthcare provider, a flagship university come together, created a joint venture. They're out there trying to help the industry in general, working with that. Hopefully we can get the word out some more, get you guys working with some more industrial partners and start doing a lot more of that kind of stuff. I love the fact that you use the word organoid. I thought that was pretty cool. I think that's whenever I do a podcast, I want to hear something new from that. So that's a good one for me for today. But this has been the Driving the Business of Science podcast. I really want to thank Dr. Joe Jerry for being with us today, giving us the insight in PVLSI, HistoSpring, and UMass Amherst. And thank you, Joe. Have a great day. Thank you, everybody. Take care.
Thank you. Bye.